Good morning children, continuation of DNA fingerprinting. How to separate normal DNA from that of the sad DNA. This is done with the help of density gradient centrifugation. The major peak will be formed of bulk DNA and the smaller pieces will be that of the sad DNA. Sad DNA means the repetitive sequences of the DNA which forms the large portion of the human genome. Also they show polymorphism. Polymorphism means different morphology which will be because of the mutation, and we know that mutation in this portion is passed to the next generation, and when it is found in the large portion of generation and becomes the more than 0.01 frequency, then it is known as polymorphism, and this is the basis of the DNA fingerprinting. One thing should be made clear, that every tissue in an individual, be a saliva, be sperm or hair or bone, they show same polymorphism. Take an example of the murder scene. So all the chromosomes are studied. I have taken chromosome number 1. This is the homologous pair. Murderers, 5 repetitive sequence, 2. Suspect 1, 8 and 6. Suspect 2, 5 and 2. So here very clearly we can see that murder has been done by this person. But it is not very easy because it cannot be seen. Also, the methodology will be complex. It consists of isolation of DNA. First step. Here what happens, as the chromosomes are present in the nucleus and that is present in the cell, so different varieties of enzymes will be used to break down, like lipase, proteases, RNase, amylase. Then with the help of chilled ethanol, we will separate DNA. Whole of the DNA is not required. We only require sad DNA for DNA fingerprinting. So restriction endonuclease enzyme will be added to separate that. So now the pieces will be transferred to for the next process, that is known as gel electrophoresis. Gel consists of agarose gel. This is anode, cathode. DNA is negatively charged, so the pieces will be attracted towards anode, the smallest, the farthest. Okay, still we cannot see this. So these pieces will be transferred to the nitrocellulose paper, and this process is called Southern blotting. Now next step is radioactive VNTR probe. What is this? A complementary to the sequences, like 80 sequences there. So TA sequence will be added and which will attach to the 80 and a hybrid will be formed. Rest will be washed away. And this last process will be autoradiography, which will be done with the help of X-ray, and here it will be visible. The sequences, the pieces, it will be easily visible. Now the thing is that when a very small piece of portion of a tissue is found at the murder place, then how to complete the experiment? Before doing this methodology, polymerase chain reaction PCR is done. So from one cell many copies will be made and then this methodology will be followed.